Koto. My name is Linn Hoffmann. I'm a marine biologist, but I work here at the botany department at the University of Otago. And my background is in phytoplankton ecophysiology. So I work with marine algae, these unicellular little algae that float in the open ocean. And I work mainly in open ocean regions. And for the last probably 15 years, I've been working quite a bit on ocean acidification. I do a lot of other things as well, and I'm mainly interested in how environmental stresses influence phytoplankton community composition and phytoplankton productivity. Phytoplankton are absolutely critically important for the marine ecosystem because they are the base of the marine open ocean food chain. So any change in the ocean chemistry or in ocean temperatures or in the ecology of the ocean that impacts phytoplankton will have impacts on the whole marine food chain. So when it comes to ocean acidification, there are several things how ocean acidification can impact marine phytoplankton. First of all, we have a few of these unicellular algae that are calcifying. And all calcifying organisms really struggle under ocean acidification. So with intensifying ocean acidification, it's harder for them to build their calcium carbonate structures, and the existing calcium carbonate structures start to dissolve. So the coccolithophores, these are the phytoplankton that calcify. They have little calcium carbonate scales that cover the whole cell. They start to dissolve, and it's starting to create problems for them. The other algae that don't calcify, some of them actually benefit from ocean acidification because ocean acidification means that there's more CO2 available for them to photosynthesize. So some of them take over, and what we've been seeing in some of the studies is that particularly toxic species seem to start to increase under ocean acidification, or we have a massive increase in certain species that start to smother everything else. So as part of my research, what we've been doing is we've been working at volcanic vent sites. So those are natural laboratories that are ideally suited for us to work under future ocean conditions where we have very high PCO2, so very low pH. So worldwide there are a few of these volcanic vent sites where CO2 is bubbling out of the seafloor through this volcanic activity, and we can use them as natural laboratories. So for example, we've been working in Japan. There's also White Island here. Fakari White Island is also an active volcanic vent site where CO2 is coming out of the seafloor, so we've been working there as well. The situation with Fakari White Island is that there are also a lot of other chemicals coming out, so it's not just the pure CO2 effect that we're seeing there. So, but for our ocean acidification studies, we did work at a vent site in Japan, and we were interested in how the phytoplankton that are first settling on surfaces, so we have biofilms that consist of bacteria but also of a lot of phytoplankton species that grow on those surfaces, how they are impacted by future ocean conditions and by ocean acidification. So biofilms are very important for settlement, for larvae that settle on those surfaces. So the biofilm community gives out chemical cues for larvae to settle on those surfaces. So they need the right composition of species there. And we were interested in looking at if ocean acidification changes that. So we did several different experiments, both at the vent site in Japan, but also in Dominica. And what we're seeing everywhere is that indeed ocean acidification is completely changing the community composition of those biofilms. And what we're also seeing that we have an increase in those potentially toxic species. We can't quite say what that exactly means for settlement of larvae in those areas, but we've also done experiments here with the Department of Marine Science. So Nadjesta Espinel, one of the former PhD students in marine science, she was doing similar experiments here in the lab, where she took water from the Otago Harbor and incubated it under different pH conditions for a long time and looked at the biofilm community on settlement plates. And she did also find some very significant changes. But she went one step further. She also looked at how these different communities were then affecting the settlement of larvae. And she also saw that there was a big impact of settlement with pH and with the changing biofilm community. So we know that there are definitely impacts, and we've done similar experiments in, as I said, in Japan, but also in Dominica. And the overall similarity is that we're seeing more and more changes in the community composition, and we see less of those calcifying algae and more of potentially toxic algae. But we can't really say for sure yet what the impact on the whole ecosystem will be. But it's quite worrying to know that just a slight change in PCO2 has such massive impact on these biofilm communities.